Hello, today we're going to be learning about conjugation chemistry from the point of view of how it's used in bioanalytical chemistry. Bioconjugation chemistry is a technique that is used to conjugate two molecules together, especially when one of them is a biomolecule. This can be, for example, to conjugate proteins, lipids, carbohydrates, or nucleic acids such as DNA or RNA to other useful molecules. Can you think of any examples, perhaps from your own research or that you've seen in the literature where you might have had a biomolecule conjugated to another? Some examples that I can think of are attaching a label for the purpose of analytical detection. For example, it's very common to conjugate fluorophores to proteins or nucleic acids so that they can be visualized on a fluorimeter or on a fluorescence microscope. It's also common to conjugate entire enzymes to other molecules in order to detect them in immunoassays or via other enzymatic bioassays. An entire other class of bioconjugation is the linkage of drugs to carrier molecules such as antibodies. And this has developed into an entire field of targeted drug delivery. It's very common for cancer immunotherapies, for example, to attach a drug to an antibody that targets a cancer cell. And so there's a great deal of interest in the chemistries that will create strong linkages and pure products. It's also common to label entire cells, usually by labeling proteins or carbohydrates on the surface of the cell, but sometimes also within the cytoplasm or even the nucleic acids in the nucleus. So as you can see, bioconjugation is very common. It's an important type of chemistry, and it's important that we know some of the basics so that we can create and use reagents wisely. So let's think about what would it take to have their perfect bioconjugation reaction. Maybe take a minute to pause the video and try to jot down maybe 3 to 4 properties that you would like to see in a reaction if you were going to design your perfect way to label a protein or a nucleic acid that you were interested in. So I'll wait, pause the video. Okay, so let's think about some properties and see how many you thought of. The first one that I can think of is that you would want it to be fast. And in particular, we need it to be fast at very low concentrations of these molecules, in particular, less than micromolar, because often that is all that we have available to us when we're working with proteins. We can't be operating at molar concentrations of protein to drive the kinetics quickly. And so we need things with very rapid rate constants. We would also like to generate pure products ideally, meaning that if you react your protein with a particular fluorophore or label, we would love it if every copy of the protein was labeled and every copy of the fluorophore was used up. You can imagine that is rare and in fact usually impossible. We would like it to preserve the integrity of our biomolecules. This means that the reactions need to be carried out under physiological conditions, particularly for pH, temperature, and also the salt concentration in solution to keep our biomolecules properly folded. We would like to be able to target particular places in the large biomolecule. For example, we would love to be able to put a fluorophore in one particular place on a protein, and so we call this property selectivity to the target site, or we might be saying we'd like to be able to target a particular functional group, such as amines, for example, without reacting with thiols. Importantly, in case we are going to do more than one type of reaction, or if we have more than one potential site for functionalization, we would like to be able to target one type of reactive site without reacting with others. And so this property is called orthogonality. It's when you have one reaction that doesn't interfere with the reagents used in another reaction. And so we'd love for all of our bioconjugation reactions to be orthogonal. We would like to be able to do this even in the presence of a complex matrix. For example, maybe I want to be able to label one protein that's in a cell without labeling all the other proteins. You can imagine that's very difficult considering that they all share the same amino acids. In terms of practical considerations, we'd like to be able to use reagents that we can obtain easily, or at least prepare them easily. We'd like them to be inexpensive. We would like them to be stable, for example, not subject to hydrolysis or other types of reactions that would break them down during storage. And we would like them to be soluble in water, so we can't be using reagents that require dissolution in things like hexane or oil, because then it won't be compatible with our biomolecules, at least if we want them to stay folded. And we'd like them to be non-toxic. So you can see this is a lot of properties for an ideal reaction. And the reality is that there are few, if any, chemistries that satisfy all of these criteria, and we're always having to make compromises. Luckily, though, there are quite a few standard reactions that provide at least some of these properties, and we're going to learn about those today. There's also a great number of cutting-edge bioconjugation chemistries that are in development at many laboratories around the world, including here at the University of Virginia, and you can read up on those if you're interested. All right, so having thought about the properties of a reaction, let's now think about where we're going to react. And here we really are constrained by the fact that most biomolecules contain only a few basic units. So for example, if we think about proteins, there's only 20 amino acids found in most proteins, right? These are the canonical amino acids. That means there are very few functional groups to choose from. The amide backbone of the protein is fairly stable and mostly non-reactive, unless you're under extreme conditions of pH. And so we're really just looking at the N and C terminus and the side chains of the amino acids. What that means is that we're mostly limited to just two nucleophiles. Those are amines and sulfohydryls, and two electrophiles, or sites for nucleophilic attack. Those are carboxylic acids and carbonyls. The carbonyls even aren't very commonly reactive. We kind of have to go out of our way to do that. So what I'm going to do now is walk you through each of these four common sites, the two nucleophiles, and the two electrophiles. Let's start with the sulfohydryls. So these are sometimes called thiols. You can see that this is a group that has two lone pairs on the sulfur atom, and these are commonly found in the side chains of cysteine. This is the most nucleophilic side chain present in your typical protein or other biomolecule, but they are very rare. And in fact, it's even rarer that they are in this reactive thiol form, because usually they've been oxidized to form disulfides, where the two sulfohydryls have reacted together, and that would have to be reduced in order to make it available to react. So to give you a sense of how rare this is, a common protein often used as standard or as a blocking agent in bioassays is bovine serum albumin. And this is a protein with over 500 amino acids, and it's only got one free cysteine to react. So these are rare, but highly reactive and useful as a target. Much more common are the primary amines. So here we have one lone pair, which is serving as a nucleophile. And these are found all over the protein, in one place at the N terminus, but also on the side chains of lysines. And because lysines make up 6% of most proteins on average, there are about a dozen of them, for example, on a typical antibody. And so that means that we have a lot of potential sites to attack. Another advantage of amines is that because they are protonated at physiological pH due to their pKa being around 9.4 when they're on the side chain of a lysine, because they're protonated, they tend to be outward facing, meaning they're on the surface of the protein facing out into the aqueous medium and therefore highly accessible for reaction. Now, the downside is that the protonated form is a poor nucleophile. And so we have to kind of balance our choice of pH for this reaction, trying to maybe bump up a little bit closer to the pKa, perhaps using pHs closer to 8 or 8.5 if we want to try to make those lysine side chains be reactive. So primary means a great nucleophile for use in bioconjugation chemistry, but you do have to think about your pH. And you have to think about the fact that there's a lot of them on the protein, and potentially, if you label too many of them, you risk altering its functionality. All right, let us turn now to our electrophiles. There are two main types of electrophiles that we usually would think about. The more common is the carboxylic acid. So this is a great location for nucleophilic attack. And this is really primarily located at the C-terminus of any protein or polypeptide, as well as at the side chains of aspartic acid and glutamic acid. Like the primary amines, these tend to be outward facing, in this case because they carry a negative charge, and so they're best solubilized by the aqueous media. And these are often used if you are using your label already has an amine on it, then you would use it to attack a carboxylic acid. Because these are abundant, this is another pretty common target. All right, so the last category, which is kind of less commonly used, is the aldehydes and ketones. And that's because these really aren't present on the side chains of common amino acids. They're not present in a reactive location on most nucleic acids, but we can create them through oxidation of polysaccharides. And as a result, sometimes this is a strategy that is used to label sites of glycosylation on the sides of proteins. So it's worth knowing that this is an option. And you may be in research where you do this all the time. All right, so here we need a little brief review of organic chemistry because this comes up quite a bit when we're thinking about bioconjugation chemistry. And this is this idea of how you would attack a carbonyl carbon with a nucleophile. So let's just recall if we have our carbonyl carbon, it's attached to, say, the protein that we'll call R, and maybe we have another carbon side chain over here that we'll call R prime. Okay, so here's our carbon. So this could be attacked if you have a nucleophile. The nucleophile, for example, might be an amine. So let's say that we have a primary amine, and this will be R2. Okay, so we've got R1, R2, and then an R prime. All right, so here's our nucleophile. We've got our lone pair, and this can come in and attack at that carbonyl carbon. And then you bump up, if we do a little bit of arrow pushing, we can bump up the electrons onto the oxygen here, and then that will eventually, there's a tetrahedral intermediate, and then that comes back down, and eventually, if this part is a good leaving group, then this will leave. So whether or not that's a big leaving group is a big part of the design of the chemistries that we're going to be talking about. But if it is, then what you end up with is an amide bond, which I will sketch out here. Here's our R1, and now it is linked to that amine that we started with, and we've got it now linked to R2, and what left as a byproduct was the protonated version of that R prime group. Okay, so again, this only works if the if the R prime group is a good leaving group in its alkoxy form. Okay, so what we want to remember here is that our nucleophile attacked at the carbonyl carbon, which was quite electrophilic, and that we end up, if we have a good leaving group, we end up with an amide bond here, for example, if this was an amine. Okay? All right, so hopefully that's a useful review. For some of you, you're thinking, of course, I know this. I just took organic chemistry, and others of you might not have taken it in 10 years. So that's why I take the time to do that. All right, so just to kind of wrap up, here's a little self-quiz for you. I have two questions. The first question is for you to rank sulfohydrals and primary amines by their nucleophilicity under typical bioconjugation conditions and justify your answer. So which one is more nucleophilic and why under these typical conditions? And the second question is, why are lysine side chains and N-terminal amines often good targets without having to denature the protein? So pause the video, take a moment, answer the questions, and then I will show you the answers. All right, so for the first one, hopefully you remember that the sulfohydrals are far more nucleophilic than the amines. This is both because they are more polarizable and have that extra lone pair, but also because the amines are partially protonated near physiological pHs and thus are in a less nucleophilic state. So there's kind of two reasons why the amines are not quite as nucleophilic under these conditions as the sulfohydrals. Regarding why amines are good targets anyway, it's because they tend to be outward facing on the surface of the protein because of that positive charge state that they're in part of the time. And so we don't need to denature them or reduce anything like we would with the sulfohydrals just to get them exposed. All right, so hopefully you now know the basics of bioconjugation chemistry, including the four major sites that we tend to target, and this review of nucleophilic attack. I hope this was useful. Thanks.